need a little bit of time to write because I just write and then I just moving quickly, right? Okay, there is another important thing under the reproduction. Yeah, the speed is okay. Good. That keep up the track. Yeah, I mean you can understand that the syllabus is quite large, right? So it's very difficult to cover up the syllabus. I think if you ask the student, those who finish A level, I'm sure that 90% student they cannot cover the syllabus. But at least one student is going for one subject, at least for three or four teachers. One is going maths class, one is privately hiring tutors, and another class like that. They all trying to find the methods to handle this exam. I'm not, I don't know about chemistry and physics to comment because I'm not doing it. But the biology, the biggest challenge is the amount of data you have to remember. Not only that, you have to use the right word in the mark to get the marks from the marking scheme. So if you write something else, that word is not in a marking scheme, you don't get the marks. That means you have to remember all contents to get the marks. That is the challenge, right? On the other hand, if you look, because GCE A level is only doing in Sri Lanka, but other countries, what are their exams and how they go to the medical faculties? Is it same? They have sometimes even bigger challenge. The bigger challenge is, for example, let's say UK, England, they also like student like you. They go to school. They have to finish their high school. They have to pass their high school exam. But the good thing is, their life is less stressful because their high school exams are not harder as this. So it is something like you have to really remember biology, everything, as in the resource book. Then you have to use this thing in the exam. But they have other exam. They are much relaxed. They are not based on remembering things by heart. They are based on the logical, analytical side of the students. It has maths, statistics, and some kind of case studies, practical, like how it's really happening things, and for that what they have to write like, not theory like this. But the challenge again they face when they want to do a medicine, they have another exam, competitive exam, like this IQ level testing and analytical testing like exam they have to pass. They are very tough. It's nothing to do with knowledge, the theory. It is something to do with the timing and how you handle this exam. Okay, having said that, one important thing, we should pace this unit 2 test. the more the question you do, it's easy for you to get the marks. Unfortunately, uh, there is not much question banks because recent syllabus changed from 2018, and it comes to the exam, the new syllabus, we have not much papers, right? So you can use I think 19, 20, 21, 21, 22, these papers, let's say maximum 4 years papers. So new syllabus, you have 4 years papers, but you can also use old syllabus. Some of the questions, it's not change everything, but some removed, some added. So old syllabus you can also use. And I think someone asked Chitsara, can't remember, Shashikala, maybe you ask about the questions like this.
Yes, sir.
Yeah, I mean, you can buy these books. They published by 2000. I think they have 2002, even old 2002 around, 2021 around we have, and mostly 2009 and 2021 is available. And there is another publisher. They are doing classified questions from the old syllabus. So classified, the unit-wise classified questions are there, but there tiny errors are there. When they're typesetting, they have some issues. Small errors are there, but doesn't matter. It's not 100% perfect. You can use this material, and then there's another component called model uh, questions and model like past exam in the schools like things. Well, there is a problem because when the exam paper uh, is making, these papers are made by at least several people. Like when we take unit 1 to unit 10, the professors who are experts in a certain area, let's say plant, then the plant professors in agriculture faculties and professors in science faculties, they take care of making question from that one. So they make unit 4 questions. And then unit 5, professors in medical faculties and other faculties, they make these questions. Like that, only different peoples are making questions. After that, they get together and they discuss each questions, the whether they are valid, they are okay, the student can understand the question, the language is okay, this in the syllabus, all these things they discuss. Even that you can notice there are errors in the past papers, there are mistakes. So that's how they make the past papers. But the model questions and what is available, they are mostly not standard. But some of the schools, they have actually bit the better ones, so you cannot rely on the older schools. Some of the schools, they have some good papers, model papers. And what as a teachers we can do individually, for example, this test, I have questions from the past papers, I have question from different exams, not GCA level, that similar exams, and also I made few questions by myself. I mean, I can't invest making the questions always, so some questions are there. So there are 50 MCQs, and you have 2 hours to finish. Time management is also important. So you have to learn time management from the beginning, reading questions, quickly answer, these kind of a thing. As a result, what I proposed, I can send the link and then you can answer and send me within 2 hours. You have to submit, it's the computer screen, everything is there. You just select the right answer and then submit the test within 2 hours. So if we cannot do at same time, For example, if you have something else, but I can send you another time. Let's say Saturday you can't do it, then you can do it Sunday. Then I can send the link Sunday. When you get the links, then you start and submit within the 2 hours. So 2 hours mean it's okay to have a little bit of plus or minus some minutes. It's not exactly 2 hours. I don't want, but within 2 hours you have to do these MCQs because timing is very important. Because many students beginning they don't consider the importance of timing because after if you don't consider timing in the exam, you face some problem, you can't finish your exam, you can finish your questions. So this is also a very important factor in the exam, the timing, right? We have to learn handle time from the beginning. So I think I will set this one 24th. If anyone cannot, let me check. 24th at 7 o'clock, I think. Yeah. 2 hours with 50 MCQ, 7 to 9, 24th. If someone cannot do this at this time, let me know, then I will fix another time, right? So this, I mean, just study the unit 2 because we finished it long ago and you have enough time. So we do this test. If you can't do it, then we can have it another time. I can send another day a link, then you can do it. Okay. Yes, binary fission. There is a situation we can see like this: two bacterial cells, they fuse together through this structure I mentioned called pili, tiny cytoplasmic projections, just like cilia, and they join together like this. Some people trying to consider this is as a sexual reproduction, but it's not a sexual reproduction because they don't make a new cell after this. But why this important? They can exchange their genetic materials, right? Fusion of two cells with pili. Fusion of two bacterial cells with pili is called conjugation. Conjugation. It's called conjugation. Now, this is something very important thing. In this method, the plasmid small round-shaped DNA molecules moving to the other side. Used to exchange DNA as plasmid. Used to exchange DNA as plasmid. So we have to talk about this in the future because some of the characters developed by one bacteria can pass to the other bacteria. So they use this method to pass some characters they develop which is useful to live, pass to the other bacteria. Because we know that meiosis, genetic material mixed up, and this is important for the survival of the organisms. So when the new organism when new organism formed, so they get different genes. This is important for the variation. So this is important in the future lessons. The conjugation is important to exchange the DNA material. So within still this is within the resource book under bacteria. You can see they use this conjugation method, right? Okay, that's all. Okay, just recap everything quickly. They have they can exist in colonies like this, colonial. They can exist as a pigment, filaments, filamentous or colonial, and they have a cell wall. The cell wall is made of this structure called proteoglycan, and then some has less proteoglycan, gram negative, and they have some kind of other polysaccharides with lipids. This is the appearance. Gram positive look a little bit of purple, and this look the pink color, gram negative. And the flagella is important structure of the bacteria. It used to move, but the flagellar structure is not same as eukaryotic. So they lack 9+2 and also cell membrane around it. So this is again unit 2. They have this 9+2 microtubule arrangement, and this is lack in the prokaryotes. And we discuss these three words: obligate anaerobes, obligate aerobes, facultative anaerobes, nutrition, autotrophs, and autotrophs. So they can also fix nitrogen, especially in plants. They can live in these roots, and they can make some kind of a nodules, and they can fix nitrogen. Cyanobacteria looks like this. So they make a filamentous appearance, and they are green color because they are doing photosynthesis. Okay, now we are coming to the next one, domain archaea. It looks very similar. Single cells, no nucleus, no membrane bound organelles, cell wall. It looks similar as bacteria, but we're going to find some of the difference. I think one of the biggest difference, which only unique, special to archaea is, they don't have this peptidoglycan, right? So peptidoglycan is only for bacteria. So I clearly mentioned peptidoglycan is specific to bacteria. So what is most special then? That means this feature is not in bacteria, this feature is not in eukarya. Bacteria is a proteoglycan not in archaea, not in eukarya. The most special molecular feature is they have branched phospholipid in cell membrane. This is the most important feature of this archaea. And there's another one similar to this, but I think resource book is not used, it's not mentioned. It's not that, I mean, important as it not mentioned here. If you remember the phospholipid molecule, there is a glycerol, and then there is a phosphate group, and there's a two fatty acids, fatty acid 1 and fatty acid 2. If you look the triglyceride phospholipid, we learn this has ester bond. So ester bond is C double O. This side is O with a carbon. So O and then carbon. So this part represents the fatty acid. This represents the glycerol. And this bond is called ester bond. But here the bond is, it's not an ester bond. It is, it's called ether bond. Okay, but this is not in resource book, right? NRB is something not in resource book. Just give me 1 second. You can copy this down, right? Right. This is something additional thing, but in case, I mean, as it is going very close to this one, right? Again, branch phospholipid. So branch phospholipid means this fatty acid is, they make branches like this. Okay, this is fatty acid. This is glycerol, and this is phosphate group. So fatty acid has a branch, is one of the most important character of this archaea. Okay. Right, other things not related to the molecules, but their habitat, the place they are live. The place organism live is the habitat, and they live in a very extreme conditions like salt. When there's too much salt, they can live, called halophiles. Halophiles, and other one is temperature. They call thermophile. Thermophiles, right? This is halophiles. The high temperature in the sense something like more than 70, 80 Celsius degree. Okay, we can understand something. How can they live in 70, 80 Celsius degree? But we don't know at the moment how the structure of their molecules, because we know that enzymes denatured at this temperature, protein denatured. So even high salt condition, again osmotic effect, they can die. So they might have more than special molecular characteristics, special molecular differences, but at the moment we discuss only a branch phospholipid is the one special characteristics of archaea. And some of the examples given, Methanococcus and thermococcus. The group of bacteria come under, the names are given, Methanococcus and Thermococcus. So Thermococcus, as name says, thermo, they live in extreme high temperature like volcanic areas, hot springs like these places. And Methanococcus, they can live in extreme methane-rich environment. This is high temperature. Methane-rich or high temperature, this one, right? Okay, next domain eukarya, right? I don't want actually there's no point actually learning characteristics of eukarya, because if you recall what you learn under the unit 2, the cell structures, so we learn everything about eukarya. This molecule and cellular level we have learned. So unit 2, everything is about that. Then we learn a little bit about the prokaryote, but we haven't learned about archaea. Domain eukarya, we'll leave it because you know all these things. But we have another important lesson. Frequently asking the questions, the difference between three domains. As in resource book, you see a big table which specify difference of these three domains. I think we focus to that one because questions based on that table most of the time. So they can ask something from the table always for the question. So we try to learn comparison of these three groups. archaea, bacteria, and eukarya. Okay, I think we discussed main difference. That means bacteria has peptidoglycan. Archaea has branched phospholipid. Otherwise, archaea and bacteria are prokaryotes. This is eukaryotes. Okay, next. So genetic composition differences. Now this is important. Okay, let's take first one, histone. So histone, you have learned before, not much, but just briefly you have learned about histone. Maybe at least you heard the word histone. Yeah, in cell cycle you learn, as also you learn from the chromosomes, I think. Yes, histone you learn. But you're going to learn histone in molecular biology. So if you look bacteria, they don't. Eukarya, yes. And archaea, some. Some archaea, they have this histone. So what is histone? Histone is a protein. So if you want to learn more about histone, we can learn what you have to learn in molecular biology. Because anyway, you have to learn this thing in molecular biology. Right? Now, it is like this. The DNA molecule of a cell, if you take a cell, the nucleus, this DNA molecule length is close to 2 meter. One cell, right? Length is DNA length is 2 meter in one cell. Okay, I think in few minutes we have to close this session, and we can start the digestion we have to start the digestion, right? Keep in mind, I just show you okay. So these are the few things where they live in extreme salty condition, extreme hot conditions, and extreme methane condition, like in stomach of the cows and some animals. They use this fermentation method. They live in methanococcus like bacteria. Okay, right. It's like this, the challenge. Now, this is a tennis ball, and I think if you get a string which is around 30 kilometer long, so you have to pack this string into this tennis ball. This represents the nucleus. Tennis ball represents the nucleus. DNA length is this strings. Now you can understand the challenge. 30 kilometer, any tiny strings or thread, you have to pack into a tennis ball. The same challenge is there because DNA molecule is 2 meter. This may be probably 10 micrometer, even less than 10 micrometer. Length of the size of the nucleus is 10 micrometer. Now, 2 meter DNA molecule has to pack into 10 micrometer space. So this challenge is overcome by the histone. Histone is doing this. Histone compress DNA molecule to make it very smaller, right? So you can consider histone is DNA wrapping molecule. I think we can wind up, stop here at this point. So histone is DNA wrapping molecule. It's a protein, it helps to wrap the DNA. So DNA wrapping is coming in molecular biology part. So we can briefly look next time that one, because you're going to learn in detail in molecular biology. We will learn next week again this section. So in the next coming up section, we move to the new lesson, digestion. So it's a DNA wrapping molecule. Absence in bacteria and